Glycolysis begins as glucose enters the cell and is phosphorylated, which transfers a phosphate group from ATP to the sugar. Renamed glucose 6-phosphate. It is rearranged to convert to its isomer, fructose 6-phosphate. This transfers a phosphate group from ATP to the sugar, investing another molecule of ATP into glycolysis. This increases the ATP molecule debit to 2. The sugar is now ready to be split in half. Fructose 6-biphosphate is then broken into two different sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. These sugars are isomers of each other. This is a reversible conversion, and although it never quite reaches equilibrium, it usually pulls in the direction of glyceraldehyde 3-phosphate to continue into the energy payoff phase. Upon entering the energy payoff phase, glyceraldehyde 3-phosphate gains another phosphate and is oxidized by the transfer of electrons NH+ to NAD+, forming NADH. It is now called 1,3-biphosphoglycerate. Now, the phosphate group added in the previous step is transferred to ADP to form ATP, renaming the molecule 3-phosphoglycerate. Now the enzyme relocates the remaining phosphate group, creating 2-phosphoglycerate. A double bond is now formed in the substrate by extracting a water molecule, yielding phosphophenolpyruvate, or PEP. The electrons of the substrate are rearranged so that the remaining phosphate bond becomes very unstable, preparing for the last reaction. The last reaction of glycolysis produces more ATP by transferring the phosphate group from PEP to ADP. This step occurs twice for each glucose molecule, increasing the output of the glucose to 2 ATPs and 2 molecules of pyruvate. The molecules of pyruvate are now ready to enter the second step of cellular respiration, entitled the Krebs cycle.